Hey guys, my name is Max and welcome to Simple Biology. Today we're going to be talking about passive transport. Now passive transport is basically a mechanism by which the cell can take materials into it or send materials out of it. It's used for a variety of different compounds that the cell needs and that the cell wants to get rid of. So obviously passive transport is going to be hugely prevalent when we talk about the cell. And so that's why we're going to be talking about that today. Now, the first thing you need to understand when we talk about passive transport is that it relies on a process called diffusion. You've probably heard of this before, but essentially in diffusion, particles will flow down a concentration gradient. This means that when particles are more concentrated in one area, the diffusion will drive them to reach an equilibrium where they're equally concentrated in both areas, throughout an area. Like I was saying, this is an equilibrium, but it's specifically a dynamic equilibrium. Basically, this means that when it reaches this equilibrium, there will still be particles going in and out of the area. Not every particle will just be frozen in place. That's a common misconception when we talk about diffusion. Basically, just kind of an overview of this process. All of the particles are moving just by nature of their chemistry, and eventually one of them will be moving in just the right manner to reach across somewhere. These two pictures show diffusion across the membrane. So this might be that it is moving just in the right manner to cross the membrane. By the time it reaches equilibrium, there's an equal number of particles moving in both directions, so there's no net movement across the membrane. The last important thing to understand is that particles only diffuse down their own concentration gradient rather than on the overall concentration gradient. If there's a lot of one particle in one area, but even more of another type of particle in the other area, the first particle will still travel into the second area, even though it's a higher concentration of solutes, just in general. Lastly, you may know that this whole process is driven by the second law of thermodynamics, which says that all physical processes will decrease the order of the universe. This is a very obvious example of that. Here you have a very ordered environment, and here there's less order because everything's just mixed together. So this whole thing is governed by that law. There's a special case of diffusion when we talk about membranes in specific, and this is called osmosis. Now, osmosis is the diffusion of water across a membrane, specifically a membrane that is called semi-permeable. In other words, it will allow water across, but not certain other components, such as these solutes that you see in this picture. Semi-permeable, the word doesn't necessarily mean with water. Semi-permeable means that it's permeable to some substances, but not others. In this case, with osmosis, we're going to be talking about water. So, as you can see, this picture shows the water is at a perfectly even level, but one has a higher concentration of solutes than on the other. Osmosis will allow water to travel into the area with a higher solute concentration, thus raising this water level and lowering this water level, so that the solute concentrations are more equal. Obviously, this opposes the force of gravity, but because it equalizes that concentration, which diffusion will want to do, this process will occur. So that's how osmosis works. Now, there's another interesting thing with osmosis when we talk about cells. Specifically with cells, you have two environments, the area inside the cell and the area outside of the cell. Tonicity is the relative solute concentration from inside and outside of the cell. There's three ways we could have this. It's hypotonic, isotonic, and hypertonic. Hypotonic means that the solute concentration inside the cell is greater than outside of the cell. In other words, water will want to travel by osmosis into the cell, thus making the cell swell up larger and, in some cases, lyse or break open. An isotonic environment is one in which the solute concentrations are equal inside the cell and outside the cell. And a hypertonic situation is where the solute concentration is actually greater outside of the cell rather than inside the cell. So this is the opposite of hypotonic. One example of a hypertonic situation would be like if you put salt on a snail. Hopefully you've never experienced that. It's a horrible thing, but it will cause those cells to shrivel down because they're in a hypertonic environment. Now the interesting thing is that plant and animal cells actually react differently to tonicity than each other. With a hypotonic environment, the plant cells will actually be normal. This is called being turgid. They're very firm. They're swollen just a little bit, but pressed up against their cell walls, which is actually the normal environment for them. On the other hand, a animal cell, represented here by like a red blood cell or something, will actually burst open if an environment is too hypotonic. On the other hand, with an isotonic environment, you have a plant cell that's flaccid, or it's just kind of floppy almost. Like if you've ever had potatoes or carrots that have just been in water too long and they're just kind of mushy, that's what a flaccid cell is like. On the other hand, for animals, they thrive in an isotonic environment. They're actually normal in an isotonic environment. That's just how they're structured. Neither one of them will actually work well in a hypertonic environment. A plant cell will become plasmolyzed, which basically means that its cell membrane has peeled off from its cell wall, and in this case, it dies and loses its function. Whereas an animal cell will become shriveled. Because it's losing the water from inside of the cell, it'll shrivel up like that. The process of osmoregulation allows a cell to alter the tonicity or to fight against it. Oftentimes, this will involve secreting certain solutes or water itself, allowing the cell to remain in its normal state, even in an environment that's not suitable for that. So that word osmoregulation essentially regulates tonicity, or osmo means the same thing as osmosis, so it regulates osmosis essentially. Now the last thing we're going to talk about is what's called facilitated diffusion. Facilitated diffusion is actually the same thing as passive transport, so now we're going to kind of get into that. This just talks about when you have transport proteins in order to allow diffusion. Now, if you remember from one of our last videos, certain particles will be allowed to cross the membrane. Specifically, small non-polar molecules will cross the membrane on their own. They won't need a transport protein. On the other hand, if you have certain other solutes or other particles, such as small ionic compounds or water, which is polar, you'll need a transport protein. Now, facilitated diffusion, as the name suggests, uses diffusion, but is facilitated in that it has a protein to allow it to occur. So, transport proteins allow diffusion to occur. There's actually two types of these transport proteins, which are channel proteins and carrier proteins. These both play essentially the same role, but carrier proteins will be more important as we go on to our next video. But essentially, the difference is that a channel protein will allow the solute or ion to just travel straight through the channel, whereas this carrier protein will actually bind with that substrate and allow it to and then that will change the shape of the protein itself, allowing the substrate or the solute to be shuttled across to the other side of the membrane. So the important thing is that the carrier protein will allow the cell greater control over the diffusion of that solute, and the channel protein will actually not allow the cell that great of control. Some of these proteins are actually gated, which means that they're closed and won't allow those solutes or those particles through in their given state, but they can be opened by specific stimuli. Some of these stimuli include different proteins or different activators changing the nature of that gate, or sometimes even a charge, like if what's called the membrane potential, if there's a charge across the membrane, if that changes in a specific way, that can allow the gated channel to open. The last little definition we need to know is that when a channel protein allows ions to travel across, it's specifically called an ion channel. And so that's all I have for passive transport. It's as simple as that.